Good evening. Thank you all for being here. Your support tonight highlights the importance of science and the incredible work happening at UTSA. We hope you enjoyed interacting with our amazing faculty and students, the future scientific leaders of tomorrow. Let's all give them a hand. Thank you. It is now my honor to introduce the new Dean of the College of Sciences, Dr. Stephanie Santarico. Under her leadership, we're excited to continue advancing research and training the next generation of scientists. Please join me in welcoming Dean Santarico.
Thank you, Taylor, and welcome everyone. Thanks for being here. Your presence to us speaks volumes. It recognizes your appreciation for the power of science and the impact that UTSA is having on the field. We are especially glad to welcome you to our inaugural Science Bytes event. So this is our first one, hopefully not our last. On behalf of the College of Sciences, I'm excited to get to introduce, I'm still learning since I am 5 weeks in. So I get to introduce and learn about some of our fantastic faculty. Our event is called From Lab to Life, the journey of a drug from discovery to healing. We're gonna begin, so I'm gonna give you a little synopsis of the three folks, and I'm gonna turn it over to the real show. So we're gonna start out with Dr. Karl Klose. He's a professor of microbiology and the Robert J. Kleberg and Helen C. Kleberg College of Sciences endowed professor and director of the South Texas Center for Emerging Infectious Diseases. His work on bacterial pathogens is leading to new vaccines and earned him recognition at the UTSA Innovation Awards. Tonight, he'll share how curiosity-driven research helps us better understand our microbiomes and why that matters. Next is Dr. Stan McHardy, Associate Professor of Chemistry, Director of the Medicinal Chemistry and Synthesis Core. He is supported by the Max and Minnie Tomerlin Velker Fund, and his lab focuses on small molecule drug discovery for cancer, infectious diseases, and neuroscience, and was recently named a Fellow of the National Academy of Inventors. And finally, we will hear from Dr. Jenny Hsieh, Chair of Neuroscience, Developmental, and Regenerative Biology. She holds the Sims Foundation Distinguished Chair in Cell Biology and serves as the founding director of UTSA's Brain Health Consortium. Her research on epilepsy, neurogenesis, and stem cells is helping us rethink what healing could look like in the future. From lab to life, that's where we're at. From lab to life isn't just a theme for tonight. It's what we strive for every day at UTSA. And together we are moving science forward as we move our students forward from lab to career. So we hope, I'm a big fan of curiosity, so we hope tonight sparks some new questions because that's where discovery begins. Thank you again for your curiosity in advance, for your support, and for being part of this journey. The future of science is bright. We are part of it, and we're grateful to be building that together. So thank you for your investment, thank you for your time, and thank you for your attention. And so with that, I'm gonna turn it over.
So we live in a world full of bacteria. Bacteria are everywhere.
And fungus too.
Don't steal my thunder. Everything that you look at, everything that you touch, everything that you sit on, everything that you put in your mouth is coated with millions and millions of bacteria. You can't see them because they're so small unless you have a microscope. But they're there and they're literally everywhere. You can find them at the bottom of the deepest ocean, at the top of the tallest mountain. You can even find them in the polar ice caps. They can live in places where there is no food, no sunlight, no oxygen. They can grow in boiling hot springs and toxic chemicals. So what are the places that bacteria like to call home is the human body. So the human body is home to over 10,000 different species of bacteria. Okay? So the bacteria that live in your human body, you have more bacterial cells than human cells. And you have far more bacterial genes in you than human genes. And what that means is that each one of you is actually more bacteria than you are human. So these microbes that coat your skin, your mouth, your urinary tract, especially your intestines, are referred to as your microbiome. They are your friends and your companions throughout your entire lives. And what that means is that these microbes have a huge impact on your health. I'm going to give you one example. So many of you probably know someone who got sick after they took antibiotics from C. diff infection or yeast infection. Okay? So what this is telling you is that your microbiome actually protects you and influences your health. So we know this about the microbiome, right? So the microbiome is influencing your health, okay? And you can imagine that a healthy microbiome leads to a healthy life. But an unhealthy microbiome is actually associated with some pretty serious conditions. So let's look at the top 10 causes of death of humans in the United States. What you'll notice is that Alzheimer's and cerebrovascular disease are right up there. And you're going to hear more about this from Dr. Hsieh. She's our third speaker for tonight. But scientists have discovered that the microbiome is involved in all of these conditions, which means that the microbial component to your health should be a major focus of scientific research to ensure that we can live long and healthy lives as we age. So what I'm here tonight to tell you is that UTSA has you covered. So the South Texas Center for Emerging Infectious Diseases, or the STCEID, is a research center here at UTSA. It is composed of 18 laboratories that study various microbes and the immune responses to them. We study bacteria, fungi, viruses, parasites, and how your microbiome and your immune system protect you and keep you healthy. So I just want to give a little shout out here because we have several faculty members from the STCEID here tonight. There's Dr. Jesus Romo, Dr. Astrid Cardona, and Dr. John Hsieh. So another important aspect of the laboratories, and Dr. Chung Yu-hung, I apologize, Chung. She's here too. She told us she wasn't going to be here, so I'm kind of not expecting her to be here. Okay, so another important aspect to the laboratories at UTSA is that they're filled with students. Not only are we doing cutting-edge research, but we're training the next generation of scientists. And every single lab at UTSA is filled with undergraduates, master's students, PhD students, and postdoctoral fellows, all focused on learning how to tackle the next problems that society faces. That could be the next pandemic, or Alzheimer's, or cancer. And I want to shout out to 2 students here in the audience right now, who are PhD students in my laboratory. That's Zach Armstrong, if you want to raise your hand, and Vita Stan. Okay, so you may wonder, how do scientists come up with new treatments and cures to the various microbial threats that we face? Well, the real answer is it all starts with basic research. You have to learn how something works in order to identify places where you can intervene. Okay? So basic research means figuring out how something works so that you can do applied research, which means then applying what you learned to come up with treatments and cures. And I'm going to give you an example. Okay? So my laboratory studies Vibrio cholera. This is the bacteria that causes the severe and potentially fatal disease cholera. The bacteria colonize the human intestine after the ingestion of contaminated water. So we asked a basic question. How do the bacteria attach to the intestinal cells in order to be able to cause disease? So through a series of genetic, biochemical, and microscopic techniques, we were able to identify the protein on the bacterial surface that specifically binds to intestinal cells. And then by studying this interaction, we were able to identify molecules that could actually block attachment. And so now we are doing applied research. We're using these molecules to try and prevent cholera. But you'll notice that it all started with basic research, just figuring out how does something work. So another important concept in successful science is collaboration. So we're lucky at UTSA because we have so many great scientists with different specialties in microbiology, neuroscience, biomedical engineering, chemistry, physics, you name it. So by working with different laboratories with complementary expertise, it propels the science to a new level that the individual labs could never achieve on their own. And I'm going to give you another example. So we are currently, over the past several decades, we have been experiencing a rise in antibiotic-resistant bacterial infections, which has led to the current crisis that we're in right now, where we're running out of antibiotics to treat disease. The bacterium, Klebsiella pneumoniae, is wreaking havoc on the healthcare system because it's resistant to almost all of the antibiotics that we currently use. So my laboratory studies basic biology and bacteria, but we don't know anything about drug development. So we're collaborating with our next speaker, Dr. McHardy, who is an expert in drug development. My lab is identifying targets in Klebsiella, and then Stan's lab is developing drugs against those same targets. And together, we will come up with new antibiotics that neither lab could achieve on their own. So, none of this, all of this scientific research is really dependent upon investment, okay? So financial support is critical to driving the progress in science to achieve all of these scientific goals. And so myself, I am eternally grateful for all of the support that my laboratory has received over the years to support the therapeutic and vaccine development against bacterial diseases that we work on in my laboratory. What I will say is that supporting and investing in science is absolutely, truly leaving a legacy of making the world a better and healthier place. So with that, I'm going to hand it over to Stan. Thank you.
So I'm going to start out by echoing one thing that Karl mentioned, and that's collaboration. And before we get into the science that we do in my lab and some of the centers we have here, I want to emphasize how important multidisciplinary collaborations are, especially in small molecule drug discovery. You can't do it alone. We can't do it without Jenny and Karl. They can't do it without chemists. We can't do it without molecular biologists, geneticists, enzymologists, right? This is all so collaborative. And so we collaborate with our colleagues that are here tonight. We collaborate with several people in San Antonio and throughout Texas. And so I'm going to show you some examples of that tonight. But before we get into that, I'd like to talk about what a small molecule drug And so I'm going to take you through a very, what we're going to call a journey of a small molecule in the body, okay? But first we're going to talk about what is a small molecule drug, okay? So I think all of us reach for the allergy medicine when the Texas cedar kicks in, right? And we take it, we feel better, and we can finally breathe, or we take a pain medication and we work too hard in the yard and our back is killing us. But have you ever thought about what actually happens when you take that drug and 2 hours later you feel much better, right? There's a really complex and systematic process that these compounds go through. But let's start out by defining a small molecule. So when you take that pill and it dissolves, you're releasing millions and millions of small little organic molecules. And these organic molecules are like a key that are going to fit a specific lock within your body. And when the key and lock come together, you're going to have a therapeutic effect. Okay? And these small molecules that are in these pills are specifically designed to go after a specific target, and they have specific properties. Okay? So what happens when we take that? Let's go through this step by step. This is kind of a fun journey. And we ask a lot of small molecules. When you consider everything they do, it's quite amazing. I love this cartoon because it captures everything that we think about in drug discovery. Okay? So when we take that pill orally, what's the first thing it does? Those small molecules are going to the stomach. They're going to be in an incredibly acidic environment, in an organ that's designed to break things down. Right? Those small molecules have to be stable. Then they're going to go through the intestine and go into our peripheral circulation. And we're going to ask that small molecule, don't you dare interact with other targets that are in the body. All the proteins and kinases and enzymes you're going to come in contact with, don't touch those. You got to go after the one we designed you for. And then it's going to find that lock, and that key's going to bind to that lock, and it's going to have a therapeutic effect, and your allergies gone. You can breathe again, or your back doesn't hurt. But we're not done with the small molecule, right? What happens next? It has to be safely metabolized and eliminated from the body, because we want drugs to be safe. We don't want side effects and things of that nature. So when you think about all these parameters that go into designing a drug molecule, it's incredibly complex, and we take in a lot of different data points simultaneously. And so what I'd like to take you through is our process of our science of how we do this. And I refer to this as the iterative cycle in drug discovery. And I think any of us that have been associated with the pharmaceutical industry or been involved in drug discovery know this cycle. So when we go to Karl and he says, here's the target we're after, we can take that target and hopefully if there's a crystal structure, some structural information, we can use computational methods to design new small molecules. We use machine learning algorithms, AI, to help that molecule fit that target. And we design the molecule the same way an architect designs a house. So you put your windows here, you want your kitchen here, you want your back porch here. That's the same thing we do with a small molecule. So we design it for a specific size, a specific shape, so that it's going to fit that lock. And when we get enough of them designed, we're ready to make them. And that's the next step in the process. So how do we make them? This is the heart and soul of synthetic chemistry and medicinal chemistry that we do in my lab. We make and break bonds between atoms to build molecules of complex structure. And we do it the same way a contractor would build your house, brick by brick, board by board. It is a stepwise process. And then once we make those molecules, now we go back to our collaborators and say, what's the activity? What do they do? And they put them in their assays and they give us that feedback. And the other thing we're going to do is we're going to take those molecules and we're going to send them to a core here. We have a UTSA called Preclinical Pharmacology Core that was developed with help from the Volcker Foundation, the Volcker Trust. And we put the molecules in there, and that group will tell us exactly the other things we need to know. How is it metabolized? Is it toxic? Does it have any cardiovascular side effects? Et cetera, et cetera. What's the half-life? All these parameters of the drugs we have to know. And we take all that data and we do analysis. And you can imagine that if you had 1,000 compounds with 30 data points per compound in an Excel spreadsheet, we get really busy very quick, right? And so we use three-dimensional data visualization programs. And what that does is it allows us to pull out the compounds that have the best properties of all those properties that we want, the best blend of properties. And then we take that data and we put it back into our design. And so this iterative cycle goes on over and over and over. And any given drug discovery program we work on, we're doing this several hundred times until we get the optimized molecule that we can then give to Jenny or someone who's running more advanced models that we can look at. So let me just take you through real quickly. I'm going to give you 3 program vignettes that we work on here at UTSA. And we work, the beauty about being a medicinal chemist is we're not tied to a therapeutic area. We can work with Jenny on neuroscience approaches. We can work with Karl on bacterial approaches. We can work on cancer approaches because the molecules are still the same. We still have the same application for different therapies. So we work in all different areas. And I'm going to give you a couple examples of these. So the first one is one of our pain programs. So we currently have 3 active pain programs. As I'm sure all of you are aware, the opioid epidemic is huge. Everyone out there is looking for novel pain approaches. So we have 2 active programs that are looking at novel pain approaches that are different targets. But I showed this program because it's pretty unique. Because this program is focused on pain that is caused by cancer therapeutics, by chemotherapeutic agents. And there are certain chemotherapeutic agents that patients take for their cancer that cause what's called hyperalgesia, or increased sensitivity to pain. And it becomes very bad because painkillers don't work and the patient becomes very sensitive to pain. And we've developed a class of compounds called protein kinase C-epsilon compounds. And these compounds completely block this effect that we can mimic in animal models. And so this is an example of a collaborative program between UTSA, UT Austin, our own Southwest Research, right, and others that are here, as well as international collaborators in London to come up with these molecules. And we're very excited, and these are now being advanced into other in vivo models. One other example is cancer. So we work on a lot of different cancer programs. We look at triple-negative breast cancer. We look at brain cancers. This particular one is focused on childhood leukemias. And this program is an example of a really cool and unique area of science that is still growing today, called PROTAC technology. And what PROTACs are is they're small molecules that basically hijack your normal cellular system to go after and degrade a specific protein that's causing the cancer. And so we're designing and making these molecules that basically fool the cell into doing this. Okay, and we can degrade the cancer target and have active cancer compounds with in vivo efficacy. So this is one program example of that. And then the third program I'll leave you with is something that we've been working on now for almost 12 years. And this is in the area of schistosomiasis. And I don't know how many of you have heard of schistosomiasis. Okay, but this is a huge tropical neglected disease. Okay, it affects over 240 million humans in sub-Saharan Africa and Asia. And believe it or not, it's second in deaths to malaria. And yet there's really no good therapies for this out there. And so some of the therapeutics that are used now only works against one of the 3 species of schistosomes, these parasites. And we've developed these compounds that act at all 3 and kill all 3 parasites. And they also have in vivo activity in animal models as well. So we're really excited about this. And this has been a collaboration with our friends over at UT Health, Texas Biomed, as well as Scripps Institute. So I showed you 3 different areas, and I told you a little bit about the drug discovery process and what we think about as medicinal chemists. And I'm going to end with a couple different messages. And I think both these are important, and you heard Karl talk about this. And that's how impactful a donor can be. Okay, but I'm going to use a specific example that I'm really proud of. And that is the Center for Innovative Drug Discovery. So, as background, for those of you maybe not familiar with the CIDD, we started the CIDD in 2012. Okay? And the idea is to bring pharmaceutical industry expertise to the academic world so that we can help wonderful academic researchers translate their basic science into real medicines, or at least precursors to medicines. All right? And viable candidates for the clinic. And we started as a small little lab out on the West Campus at UTSA. Okay? And we have been operational for the past 12 years, 13 going on 13 years. And we've grown substantially. And now we encompass 4 core facilities, which we used to just have 1 lab. We have 4 core facilities, 2 at the Health Science Center and 2 here at UTSA. We have a computer-aided drug design core, we have a high-throughput screening core, we have a medicinal chemistry core, and we have our preclinical pharmacology core. All 4 of these work synergistically together in trying to develop small molecule drugs for targets. And I want to highlight this as an impact, because what started as a small initial investment by a donor in 2012 has now, as of today, we've recognized over 110 million in grants and contract research. We've worked at over 60 drug discovery programs. We have anywhere from 12 to 15 active programs on any given year. So it's been a real success story. I think it's been a great impact for UTSA. And the thing that we didn't fully appreciate when we set up the CIDB, and Karl alluded to this earlier, is we brought all this pharmaceutical industry expertise and technology into UTSA, but we never really appreciated how big of an impact it's going to have on our students. And so what we've seen over the past 12 years is now these students are working in the CIDB side by side with our staff that are trained and experienced, and they're learning these techniques. And it's really having a direct impact on their lab and career transition. And so just as an example, in the last few years out of my lab, we've had several students go to pharmaceutical companies like Pfizer, Merck, Bristol-Myers Squibb, and some of the other top pharmaceutical companies. And so I want to leave you with that message, like Karl said. The impact not only to the cutting edge research at UTSA is huge, but the impact to our students is amazing. So thank you, and I'm going to turn it over to Jenny.
And the key to chronic diseases, the key to understanding how we age, are already locked in ourselves. We just don't have the key to unlock them. And so by having a personalized stem cell, we can begin to understand and decipher the mysteries of our own cells, unlocking the key to healthier aging and preventing diseases. So how do we make these personalized stem cells? But before I tell you that, I want to remind everyone, and we probably don't need to remind everyone, because we all have friends, maybe even family members, we all know people who are suffering from Alzheimer's and related dementia. And in fact, estimates from the Alzheimer's Association, as of last year, the number of patients with Alzheimer's and related dementia is now totaling nearly 7 million. I mean, that's 7 million people, and that number is always increasing. And so the rise of neurodegenerative disorders like Alzheimer's and also other chronic neurodegenerative disorders, cerebral vascular, diabetes, cancer, so all of these disorders, perhaps we can maybe now finally attack and have something in our arsenal to be able to prevent. So how do we harness the power of stem cells? Well, what is a stem cell? So a stem cell is the mother of all cells. It is the cell that produces all of the cells, the hundreds of millions of cells in our body. And this mother cell becomes the blood and immune cells that Karl talked about in the beginning. That's really important to defend our bodies against microbes and viruses and parasites. The mother stem cell also becomes all of the cells that make up our liver, which we're so glad to have these cells, especially if we're celebrating, like when we're upstairs in the reception area. These ever important course, the mother stem cell becomes the nerve cell in our brain and in our spinal cord, which makes us very uniquely human. But these stem cells are found during development. And so they're found as we are in our mothers, as we're becoming babies, and we run out of these stem cells when we become an adult. But now, what I'm going to share with you is that there is revolutionary technology to make personalized stem cells. But before I share with you that technology, it really starts with a fantastic team. And we've already heard Karl and Stanton talk about how we're so fortunate here at UTSA to have our graduate students and our undergraduates and our master's students in our laboratory who are so committed. Here on this slide is just some of the students in my lab who's working on some of these personalized stem cells. They couldn't be here because, for example, Hsieh is interviewing for a postdoc position today, so she's actually at Duke University, and some of my undergraduates are either in the lab or they're studying for the MCAT. But I do want to mention Dr. Chris Navarro. I want to give a shout out. He's here. He is the director of our UTSA Stem Cell Core facility. And the Stem Cell Core is where all of this exciting stem cell research is happening. And so what we can do now is we can recruit patients with chronic neurodegenerative disorders like Alzheimer's to our Stem Cell Core facility. And with a small blood sample, we could convert these blood cells into personalized stem cells. And these blood cells, when you introduce 4 genes, they forget that they're blood cells. And they, over the period of weeks, they start turning into that stem cell that was found in development. And now, in a dish, can grow into any cell type that we direct them to. So we could, for the first time, using these personalized stem cells, begin to understand how Alzheimer's might develop in the first place, or how stroke might happen, or even how cancer might happen from these personalized stem cells. And another very exciting technology is CRISPR gene editing. And this type of gene editing is similar to if you're typing on your computer and you make a mistake in a word, a typo, you can delete that mistake and enter the correct letter. So we call that word editing. So this genetic editing is like a pair of molecular scissors that scan your DNA, your genes. And if you're a patient that has a genetic mutation, it can cut out that mutation and replace it with the correct gene, a normal copy of that gene, in the personalized stem cell. And what's really exciting is that these personalized stem cells that come from the patient with a genetic mutation can be grown into a brain organoid. These are human brain tissue. They resemble human brain tissue, but in a dish. They're like a mini brain in a dish. And they have a lot of the complexity and the cell types that we might observe in a developing human brain. So these models and the technologies, personalized stem cells, CRISPR gene editing, and brain organoids, are really game changers. And the reason why they're game changers is because no 2 patients are alike. Every one of you in this room, we're all different. We're all different by our genetic makeup. Even identical twins with the same genes, but because of their different environment, their different lifestyle and experiences, in children or adolescents, they exhibit signs of difficulty focusing. They also have lower IQ, and they also have weaker short-term memory. So this suggests, at least this is the hypothesis, that maybe individual with certain Alzheimer's disease genetic risk factors, there might already be changes very early in their brains as they're developing and as their brains are functioning, forming. So now, with a brain organoid from patients with a leading genetic risk factor like ApoE4, we can begin to look at this. So these are examples of slices through brain organoids. Now, brain organoids are very tiny. They're tiny three-dimensional structures that resemble the human brain. But using a microscope, we can magnify the size of the tissue, and we can look at markers. So green is a marker of a neuron. And hopefully you can see that there's big differences, right? So on the left is a slice through a brain organoid from a healthy patient with an ApoE3. But on the right, we see a lot more staining of this marker. And the process of those brain cells seem to be more exuberant. So these data suggest that perhaps individuals with 2 copies of the ApoE4 gene, they may have faster brain development, and their brains may mature quicker. So what might this do functionally? So we can measure this. So we can measure these brain organoids using measurements of their electrical properties. And what we found is the brain organoids for a patient with Alzheimer's carrying this leading genetic risk factor, on the bottom there, they have a lot more active neurons. Their brain organoids are hyperactive. There's a lot more of those green lines, and each of those green lines is an action potential. So these data are suggesting that by recreating the brain development in a dish, we can already begin to observe early growth of the brain, and this correlates with hyperactivity. And our hypothesis is that this early brain maturation, perhaps in an Alzheimer's disease patient, may over time lead to destabilization of their neural network, ultimately leading to a decline in brain function. And so again, using these personalized stem cells and brain organoids, we could now begin to test drugs in a personalized way and be able to rapidly develop drugs and use them to help patients. So our call to action today is emphasizing how important it is to support and invest in these types of early stage studies. Also, these early stage studies many of them are really outside the box, like what I'm describing here. And again, every single major medical breakthrough came originally from basic research. And here at UTSA, it's all about collaboration. And so in order to tackle some of these most challenging, complex problems related to neurological disorders, we have established the Brain Health Consortium. And we now have over 80 researchers working across 5 different UTSA colleges using transdisciplinary approaches like the one we talked about today to do this basic research to be able to translate from lab discoveries to the clinic. So I leave you with a final thought.
Let's give a round of applause to our wonderful speakers. Thank you. And I did want to open the floor up for any questions. So if anyone in the audience has questions for our wonderful speakers, we can take them now. No pressure, but maybe a little pressure. Yes, Pat?
First, thank you for having this. This is very wonderful. I feel like I'm watching Star Trek. How do you use AI? And what do you see the future of AI helping you in your research?
Yeah, I'll tell you how we use it. So we use it by more of a machine learning aspect. So what you do is you feed a computational program data, and then it uses that data to go out and reach out and pull more information in to build a more accurate model. So we use it on the design aspect, but most of it is machine learning as opposed to AI. But it's very, very helpful in the discovery process because it allows us to look at millions of compounds at a time, as opposed to a medicinal chemist sitting in an office with a modeling set looking at one at a time. And so it increases the efficiency and decreases the time. So that's how we use it.
I was going to say, Dr. McHardy, to add to that.
Cool. Yes.
So in our field, in molecular microbiology, I work in Dr. Klose's lab. We can use AI to kind of do a lot of the back-end work as far as finding the therapeutic value. And maybe there's a certain protein that's involved, like Dr. Klose was mentioning, that's necessary for causing disease. And you can use AI to predict, small, very minute changes that we can make to that protein that thereby inhibit the bacteria and its ability to cause disease. And so AI is instrumental. It's super convenient, and particularly whenever it's used to build machines that can inform you of decisions that are otherwise kind of costly or research intensive.
So, Pat, I could mention that biologists are using AI, both neuroscientists but also stem cell biologists, to make models of the biological system. And the reason is because we may collect data, we have our system, we collect data, and we try to explain what's happening in this black box. But using that biological data, we have in parallel like a computation or an AI-based model. We can put the data in there and then use it to predict or generate new hypotheses that then we can test in the biological model. And it can be very powerful that way.
Gary, yes, please. So on the biome, right? I suppose the viral load of the biome is greater than the bacterial.
Oh, see?
I'd be interested in your comments as to the viral load on the biome.
Yeah.
This probably is greater even than the bacterial.
Yeah, that's a great observation that you have there. So the largest biological component on Earth is actually viruses and bacteria. So there's far more of those even than bacteria themselves. And so you're exactly right. If you were to look at your own microbiome, you would probably see even another level, sort of another log increase in the actual bacterial viruses that are in it. And that doesn't mean that there aren't other viruses too, of course, right? So there's viruses of your own cells, and there's viruses of fungi, there's viruses of parasites, but the bacterial viruses far exceed anything else on the Earth. It's kind of amazing. And by the way, that's where CRISPR comes from. So the whole CRISPR system comes from bacteria trying to protect themselves from this virus attack. Right? So we really kind of owe bacteria something, right? Standing ovation because of CRISPR, right? And again, another little plug for basic research. The researchers who discovered CRISPR were microbiologists, and they were just trying to figure out how the bacteria protect themselves from this onslaught of viruses that they're constantly being bathed in, right? And that led to the CRISPR revolution of editing, eukaryotic genes at will, but it all comes back to bacteria. I just want to make that clear.
personalized medicine. It seems to me that over recent years, I've heard shows or something talking about that, but then it seems like in different ways they show that it has failed. And I know we're kind of at the beginning of a medical revolution and all, but I just wonder, why so far have there been so many failures, and what exactly are the challenges that need to be overcome for personalized medicine?
I mean, that's a great question. I think the best success story is in cancer, right? Because knowing your cancer gene, doctors like Stanton and his team could design small molecules that attack your cancer specifically, and you don't have the off-target effect. In the brain, the most challenging thing is that most neurological disorders are not genetic. The ones that I mentioned, the one that I mentioned, ApoE4, there are healthy people, many healthy people with this allele that never go on to develop Alzheimer's disease. And Dr. Perry is here, and he's certainly the expert in this area. So most are what we consider sporadic or not genetic, and that's where it's the challenge is to find just targeted drugs to the receptor or the gene, because we don't. It's certainly what we call polygenic or epigenetic, but it's not a single gene. Thank you.
Anyone else? Yes, go ahead.
Last question too.
Okay, last question.
Okay, so they have genetic studies. You could swab your mouth, send them off to a company, and you'll get your genetics and how it fits drugs. And then some drugs will be fast metabolizers, some drugs will be slow metabolizer. You'll get a little flag that says you never take this drug because it's too fast, it's not going to work. If it's too slow, you might get side effects. So how much is that? Should we champion, or should we?
Yeah, so this is a great question. And we take it into consideration in drug development, and I guarantee the pharmaceutical companies are doing this right now, right? So for example, when they design a drug or they work on a drug, I'll give an example of something that is called CYP450. So we have all these enzymes in our body that oxidize things and break them down, right? They're called CYP450s. The problem and the reason you take those tests, and what we have to do as medicinal chemists, is we have to design compounds that avoid certain CYPs. And the reason is because some people express those CYPs higher, and some people express them lower. And so if you're a high expressor, and then you take a drug that's metabolized by that CYP, or even inhibit another CYP, it's going to change the drug action. So that's the basis for that. But they absolutely pay attention to it, because we don't want a compound that inhibits a certain CYP or is metabolized by a certain CYP to a certain extent, because you're going to have patients that are high expressors and low expressors of those.
So I went to that doctor, one doctor, and say, yeah, this is going to be in the future. And another doctor could say, this is garbage.
Well, I'll tell you how serious it is. They take it into account when they're recruiting people for clinical trials.
Nice.
So you do those studies because they want their studies to be accurate. And if you have someone who is a rapid metabolizer of a drug that you're making that hits that enzyme that they have, it's going to leave a data point that's off. And so yeah, they actually do those studies and they pay close attention to that. It's a really important piece.
If I could just add, because you mentioned the clinical trial, is that I forgot to mention in my talk that one of the limitations is that the one-size-fits-all drug just doesn't work. And I think we think that part of the power of this personalized science and having a more personalized model is that maybe in the future it could be an N of 1, right? And that means, like, I know of a study that's funded at Purdue that is it's a sodium channel mutation. It's a very severe epilepsy, And the researchers have, they have a foundation, they're supported by a private foundation that just funds this gene, and that it's to fund the drug, from discovery to healing, that essentially then will go back to the one patient. So it was very revolutionary, just that model.
Oh, we've got another question in the audience. Yeah.
This might be for Jenny, but are y'all looking at ways to slow down the hyperactivity or maturation process?
So the question is, are we looking at ways to slow down the hyperactivity? And the answer is yes. So in fact, this study came from this idea because some patients, but also animal models with this Alzheimer's risk factor show seizures in the early stages of their disease. And there have been studies from UCSF and Gladstone that suggests that treating patients with with this particular genetic allele with anti-seizure medication seems to slow down the progression of Alzheimer's. So this hyperactivity hypothesis of Alzheimer's is of course one of many hypotheses around Alzheimer's, but that's why we're excited by having these organoid models to begin to test that theory.
Probably have time for one more question, if anyone else in the audience has any. Okay. Well, thank you very much for attending our first Science Bites, and we hope to see you at the next one in the fall. I don't know yet, but we'll take your suggestions. Thank you. Let's give our speakers a round of applause.